Now in today's video we are going to discuss about Golgi bodies or Golgi complex or Golgi apparatus. Okay, so this is from chapter number 8 from class 11th biology. The chapter is cell, the basic unit of life, and in eukaryotic cell we are learning different organelles, and we have completed endoplasmic reticulum in our previous video. In this video we are going to discuss about Golgi bodies. Okay, so this is a complex structure. Okay, and this complex structure is made up of smooth membrane. Okay, it is not having any kind of ribosome or any other particle that is located on its membrane. It is smooth membrane structure, or you can say saccule or cisternae. Okay, saccule or cisternae means bag like structure, and it is smooth membrane, bag like structure. Now this is in form of cisternae, which means it is having a flat disc like structure. Okay, and that is parallel to each other. Okay, and this structure is located near the nucleus. Okay, it is always located near the nucleus and endoplasmic membrane. So it is near this area. Okay, wherever the Golgi body is present, that particular area is devoid of any kind of cytoplasmic structure. It means that area is excluded part of cytoplasm. It will not contain, it will contain very less structure, or you can say zero, and nothing is there, only cytoplasm, and in that area our Golgi body is present. Okay, so it will not contain any other cytoplasmic structure like mitochondria or ribosome situated near there. So it is present in very in a cytoplasmic structure where it is you can say it is a zone of exclusion. What I have written here, zone of exclusion. So wherever the Golgi apparatus is located, generally it is present near the nucleus and near the centrosome. This is a location of your Golgi complex. Now this Golgi complex is consist of stacked cisternae, which will also contain a network of tubule. You can see here I have made tubule also. So this pipe like structure, tubule is also present there, and it will contain vesicles also. Okay, and it will also contain Golgian vacuole, or you can say vacuoles like structure, bag like structure, small spherical bag like structure. It is present in Golgi ground substance or zone of exclusion. Now it was first discovered by Camillo Golgi, and in honor of him, in his honor, it was named Golgi apparatus. This structure was finally confirmed by Dalton and Felix. They confirmed this existence of Golgi apparatus or Golgi complex by observing it under the electron microscope, and therefore Golgi apparatus or Golgi complex is also named as Dalton complex, just because of Dalton. So in 1898 it was discovered first by Camillo Golgi, and before him in 1867 George has also observed this structure, but it was finally with evidence it was given by Camillo Golgi. He described it about Golgi apparatus, he gave the description, so in honor to him the name is given. Now what is Golgi apparatus structure consist of? So it is consist of cisternae, tubule, vesicle and vacuole. It is having two face that is your forming phase, that is your cis phase. Okay, so whenever, what is the function of Golgi apparatus? Its function is to receive the material, or you can say protein or any other secretory substance from endoplasmic reticulum. Okay, it is a part of endomembrane system, so it will receive anything from endoplasmic reticulum, let us suppose protein, and now this protein will enter Golgi apparatus, and from there it will be moved to any other whatever organelle it wants to get transferred. It will move there to its destination organ, to its destined organelle. Okay, and this all movement will occur in form of vesicle. Okay, so if the protein is transferred from ER, that is your endoplasmic reticulum, to Golgi bodies, it is coming in the form of vesicle, and that vesicle is known as transitional vesicle. Now this transitional vesicle will first combine with, or it will go and fuse to Golgi apparatus. Now this Golgi body consists of three parts. First is cis phase. Now this cis phase is the phase where it will receive the protein. It will receive protein. It will always present near the endoplasmic reticulum. Okay, and your trans phase is always toward the plasma membrane. So it is toward the outward. It is also known as maturing phase. Trans phase is also known as maturing phase, and cis phase is always known as forming phase. So it is said that it will, the movement of any material or anything that has to be transported will be from cis phase to the trans phase. Cis phase is generally forming phase, and trans phase is generally maturing phase. So you have to remember this point. Okay, so any transport vesicle is coming from endoplasmic reticulum, it will come from cis phase. Okay, it will come toward the cis phase. Cis phase is going to receive that material. Okay, and that will pass on to the next cisternae, and then to the next, and then to the next, and at lastly it will be passed to trans. From there, from trans cisternae, now it will form secretory vesicle, and it will be removed from here, and it will be sent to whatever the destined organelle is there. Okay, either to plasma membrane or a lysosome or any other endosome. So it depends, okay, what kind of material it is and where it has to be sent. You can also understand it like this, that Golgi complex is a kind of location where everything will come and it will get manufactured, it will get modified, and then it will be sorted and then shipped to the certain area or certain destination. Okay, next, after this, let us see what are the important function of Golgi complex. The first function is to do carbohydrate synthesis. Carbohydrate synthesis, many carbohydrate like sialic acid or galactose are synthesized over here. Next is your protein sorting. Protein are sorted, they are modified. Okay, that is their important part of protein is modified, and then it is transferred wherever it has to be transferred. Next is your transformation of membrane. Transformation of membrane, you can see this is a kind of process where the recycling of membrane will take place. Recycling of plasma membrane or endoplasmic membrane will occur. So this always happens in Golgi bodies. So it is going to receive any endoplasmic membranes portion, and that will be formed in the form of a vesicle, and it is going to utilize them. So transformation of membrane is a process where you can say the membrane is recycled, either it is of plasma membrane or it is of endoplasmic membrane. Next is glycosylation of protein and lipid. Protein and lipid, different protein and different lipids are covalently linked with carbohydrate chain. Okay, with some kind of oligosaccharide are attached to the protein and lipid. Now this process also occurs in Golgi body. Next is your many secretory product, like different hormones of endocrine system. They will contain the cell which are having large number of this your Golgi apparatus or Golgi stacks. Okay, why? Because they are, due to the presence of Golgi apparatus, they are able to form more and more secretory products. So in glandular cell, large number of Golgi stacks are observed. Next is your formation of acrosome. Acrosome is present in sperms. Okay, in sperms, at the tip of sperm there is a acrosome which will contain a certain enzyme that will help in digestion of eggs outer covering during fertilization. So that acrosome, okay, this acrosome is actually formed by Golgi bodies. So formation of acrosome occur in Golgi bodies. Vitellogenesis is actually the formation of yolk, process of formation of yolk. Okay, yolk formation is actually occur in egg cell near the Golgi bodies. Okay, Golgi body is always in center wherever the yolk formation will take place. Okay, next is your formation of plasma lemma. Formation of plasma lemma, that is your plasma membrane, or formation of your primary cell wall during the cell division of plant cell. Okay, first that cell plate is formed. During all this process, Golgi apparatus is involved. Okay, Golgi body directly helps in formation of plasma lemma or plasma membrane, primary cell wall, and also in the formation of lysosome. So this was about Golgi apparatus. In next video we will discuss about lysosome. Thank you.